Welcome back to Divan's Biology. My name is Dr. Aruna Divan and today we are going to talk about the basics of DNA replication and the enzymes that are involved in this replication. Okay, DNA is a carrier of genetic material. Now being a carrier of genetic material, DNA has two main functions. The first one is heterocatalytic function and second one is homocatalytic function. Right. Now in heterocatalytic function, DNA synthesizes materials or molecules other than itself. What can be those? Proteins and RNA. In homocatalytic function, DNA synthesizes itself or it replicates itself. So today we are going to see homocatalytic function of DNA. Right. Now in DNA replication, three mechanisms were thought of. The first one is conservative, second is dispersive, and third one is semi-conservative. Right. In conservative mechanism, the DNA, DNA is double-stranded molecule, right? Now in conservative mechanism, DNA, the parent strand of DNA or parent molecule of DNA remains as it is, means it remains conserved and a new molecule of DNA is formed. In case of dispersive, the parent DNA molecule disintegrates and two new molecules are formed. In case of semi-conservative, now as you know, DNA is double-stranded molecule. So in semi-conservative, what happens? The strands splits apart. The strands splits apart and two new complementary strands are formed. So these are the two new molecules, of which one strand is of parent strand and other one is newly synthesized. Okay, today we are going to talk about semi-conservatives because there are many evidences which prove that DNA replicates through semi-conservative method. Now replication of DNA, as we can see, it looks very simple, but this is a very complicated method. Why is it so complex? Because in your genome, there are 3 billion base pairs and all of these base pairs needs to be duplicated or needs to be copied. You have in your body trillions of cells, and if any one cell is planning to divide, then it needs to copy 3 billion base pairs correctly. Okay. For DNA replication, we know that we need to separate these two strands, right? Now you know how these two strands are connected with each other. They are connected with each other by hydrogen bonds in between them. This is a little detailed structure. Here you can see these are the nitrogenous bases and they are connected by hydrogen bonds. Now you know that A and T will bond by 2 hydrogen bonds and C and G, they will bond together by 3 hydrogen bonds. Okay, now to separate these two strands, this is first strand, this is second strand. Let us suppose this is 5 prime 3 prime direction, then this would be 3 prime and 5 prime direction. Okay, now to separate these two strands, we need to dissolve or disintegrate or break these hydrogen bonds. Okay, so when these hydrogen bonds are broken down, then it is said that these purine and pyrimidine bases attract immediately, if possible, the complementary nucleotide bases, complementary nucleotide. Okay, now let us suppose the hydrogen bond has been broken. Okay, now this strand, this is first strand, this is second one. Now this strand will form a complementary or will attract a complementary nucleotides. The complementary nucleotide will be this. Okay, then hydrogen bond will be formed. As soon as hydrogen bonds are formed, the phosphodiester bonds will be formed to make a new molecule of DNA. Similarly, this strand, this strand will form its complementary strand similarly in the same method, by the same way. Now, this looks very simple, but there are many chances of error, right? And many enzymes and proteins are needed to do this replication correctly without any mistake, as well as many enzymes are required for performing all the steps of this DNA replication, right? First of all, what is needed? First of all, the thing that is needed is to break down the hydrogen bonds, right? Now, who is going to break down this hydrogen bond? Let us form a simple structure. Here we can see the hydrogen bonds are already broken, okay, and then here we will form the structure like this, okay. Now this hydrogen bonds are already broken, but who has done this? Here hydrogen bonds are there, okay, here it's still coiled. Now, who has broken this hydrogen bonds? For this, the first enzyme that comes into action is helicase enzyme. This helicase enzyme will break down these hydrogen bonds, okay, and form a replication fork. Now, we need to form a complementary strand, right? Let us make this complementary strand with this red color. Now we need to form this complementary strand. Now who is going to add nucleotides here to form complementary strand? For that, the second enzyme that comes into action is DNA polymerase. We have actually three types of DNA polymerases: polymerase 1, 2 and 3. Here the main enzyme is DNA polymerase 3, which is going to add new nucleotides on the complementary strand. Okay, let us suppose this is 3' and this is 5'. So the complementary strand that is going to form should be in 5', 3' direction, right? Now, the special characteristic of DNA polymerase 3 is that it can add nucleotides only in 5-3- direction. It cannot add nucleotides in 3-5- direction. So this can be formed easily because this is in 5', 3' direction. What about this complementary strand? That we will see later, okay? And the second thing about DNA polymerase is that it cannot start from the scratch. What does that mean? It means this DNA polymerase 3 cannot form this complementary strand by itself. It cannot start. It can add nucleotides on 3-OH free end. Now, where is 3'OH free end? We don't have it. So, DNA polymerase 3 cannot work even on this complementary strand. So, now comes into action the third enzyme, that is primase. This primase enzyme will add a small primer here. It will add a small primer, means few nucleotide strand it will make here, right? After this strand will get 3-OH free end here. So now DNA polymerase 3 can work. Okay, now DNA polymerase 3 will start adding nucleotides here. So this will be formed easily. Now what about this strand? This strand is 3-5- direction. Now DNA polymerase 3 cannot work in this direction. It needs to work in this direction. But as the fork is forming, or as the hydrogen bonds are breaking, simultaneously DNA polymerase 3 is working. So it will form, let us suppose up till now DNA has, up till now this much is the fork formed. only these much hydrogen bonds are broken. Okay. Now this DNA polymerase 3 will work properly in this direction. But here DNA polymerase 3 needed a primer also. So in this complementary strand a primer will be added, right? When primer will be added, this will be 3-OH end here. So the nucleotides will be start added here in this direction. Now when more hydrogen bonds are broken, then what will happen? Then again DNA polymerase 3, then again one more primer will be added here. One more primer will be added here, and then DNA polymerase 3 will work again in this direction. So finally there are few small fragments formed in this strand, right? Now this is DNA polymerase 3 also needs a template. We have a template here. We also have a template here, right? So the first thing is that it needs a DNA polymerase 3 needs a template. We have it, right? Second main thing is DNA polymerase works in 5-3- direction. Third one is it needs a 3-OH free end, right? So primer is there to give it a 3-OH free end. Now this strand which is forming continuously, this strand is known as a leading strand, and this strand is known as a lagging strand. Now as this lagging strand is forming small fragments, right? So those fragments are known as Okazaki fragments. This Okazaki word is given after its discoverer. Now one more thing, one more problem. When there is a coiled thing, if there are two threads that are highly coiled with each other, if you try to open those threads from one end, obviously the other end will be having supercoiling, right? The same thing can happen here. So now who is going to solve this problem? For that, one more enzyme will come into action, and that is topoisomerase. Right? Topoisomerase. DNA gyrase is actually a kind of topoisomerase which works here. Right? This topoisomerase will form a small nicks in these hydrogen bonds so that this supercoiling is avoided or it is prevented. Okay. Now the next thing is that this topoisomerase have done its work, but now what happens if when after uncoiling of this strand, if it coils back again? This is also a possibility. So for that, few proteins known as single-stranded binding proteins. These are ring-like proteins, and they will join or attach themselves here in the replication fork so that the DNA does not recoil it back. Okay. So this is SSBP, means single-stranded binding proteins. Right. Now next what? Now next is when these two complementary strands are formed finally, let us suppose they are formed. Right. Now at the terminal end, what happens? At the terminal end of DNA, this primer is not able to sit there. So what happens? The terminal portion of the DNA is not able to replicate. If this happens continuously generation after generation, the DNA will start shortening. So there might be somebody to help this out, right? That is telomerase enzyme. This telomerase enzyme have its own template, and it will add few nucleotides so that the primer gets a space to sit on and replicate the DNA. Okay. Now when this everything is happened, then these primers are now needed to separate off because these are RNA primers. So this RNA and DNA combination doesn't work. So these primers need to be separate out. Okay, and add a DNA strand. So this will be done by DNA polymerase 1. Okay, DNA polymerase 1 will remove this primer. Now these are the nicks which are formed because these primers are being removed. So these nicks are being filled up by the enzyme DNA ligase. Okay, so what we are going to do now is we need to summarize and we need to think about what questions can be asked in the exams. For that, you need to understand and memorize all the characteristics of DNA polymerase 3. Okay, the first one is it cannot start from the scratch. It needs 3-OH free end. The second one is it moves from 5- to 3- direction only. It works from 5- to 3- direction only. It adds nucleotides in 5-3- direction. Right? It also do the proofreading. What happens if some wrong nucleotide is added? Then this DNA polymerase 3 itself will check it. It will do the proofreading. It will remove that and add the correct nucleotide there. Okay, so you need to memorize all the enzymes, proteins and their functions. Right? I hope you might have understood all this. Thank you.